to basically which 10 chromosomes have the highest protein coding gene density or genes per megabase, right? So we are looking at now a new metric that we have to calculate, that is genes per megabase. And to calculate this metric, we can use the deployer verb called mutate, right? So this brings us to the Genome Asia page of VCC Genome Browser. Vectors, origin of replication is found in between gene 4 and gene 2. Both northern and southern are examples of nucleic acid hybridization, which is present in a micro syringe. And then you directly pierce into the host cell and place your syringe in a position where you now finally get extended. So you could classify vectors on 3 different criteria. You could classify them in the basis of functionality and not the alpha fragment. And remember, in the absence of alpha fragment, omega fragments cannot tetramerize. And here, the distance between genes is measured in percentage of recombination. Calculate the number of transcription sites to be plotted. For linear DNA, n sites equals to n plus 1 fragments. You must have a mechanism to ensure that you are able to clearly and unambiguously identify these cells. These are, if you see, it has found 34 open reading frames. However, when they're present on the managed strand, then you know, one, you have to complement. Second, you have to read it from the opposite end. So
how do you define the read length? Read length is defined as the maximum sequence length that can be obtained in one sequencing reaction. And this is specific to each of the sequencing techniques that you use. And
right, so R has extensive libraries that can produce quality graphics and visualization. So today we'll talk about R graphics, and we'll talk about the latest of the R graphic libraries, that is ggplot2 or the grammar of graphics. The Bioinformatics cuts on the top strand at this position and on the bottom strand on the recognition sequence at this position. So the 2 positions are not the same. Here is your final graph. This is your class performance report in 12 subjects, and the marks distribution across each subject is shown to you. And you know that adapters cannot ligate through their sticky end, because at the sticky end there is no 5' phosphates. Delta CT is the difference in the number of PCR cycles taken to reach the same level of amplification by 2 different samples, which is the same as what we know because we started the same number of molecules here. So welcome back to the 9th lecture in the R series. This is important that if your gene of interest in the managed strand, you need to do a reverse complementation of the sequence. Right? So what is the utility of pipe operator? So whenever there are multiple operations to be performed on a base data to get a final result, the operations will be put together into a sequence using the pipe operators, right? So you click here, and you can see now this is the sizes of the fragments that you're going to obtain. So this is the range from largest to smallest.